The fact that she's still standing is a good thing. This is an indication that one does survive their PhD research on defense. And she's currently a guest researcher at the German Research Center for Environmental Health in Munich, Germany. Elaine's dissertation focused on the influence of genetic and environmental factors on the development of childhood allergic diseases and the potential role played by gene-environment interactions. Elaine is also interested in how other environmental factors such as noise and green space proximity may influence health, and is continuing to pursue this line of research as part of several international projects in Europe. So you can tour the world with research in this field. And want you to please join me in welcoming Elaine here today.
So thank you for having me here today. And as Karen said, my research for the last 4 years or so focused on the risk factors of childhood allergic rhinitis. And as I was invited here to speak today, I decided that I'd probably give an overview of what I've been doing in order to sort of give back to the school because they have given me so much over the last 4 years. So just as a brief outline, I'll be talking a little bit about what is allergic rhinitis for a few slides to give a little bit of an introduction. And then I'll briefly go over the 4 studies that I conducted as part of my PhD and to see how this all sort of fit together in the end, hopefully to give a complete story. And also touch on some of the important strengths and limitations of the research that I did, as well as the conclusions that I was able to draw. You'll see there's only 3 of them that I was able to draw, which I think is not bad for about 4 years of research. And I'll finish off with my acknowledgements. So allergic rhinitis, in its simplest form, is essentially inflammation of the nose that results in nasal obstruction, itching, or sneezing. And this is when an individual does not have a cold. It's also often accompanied by itchy eyes. And so the allergic component of allergic rhinitis implies that one is actually sensitized to an allergen. And so this is a somewhat complicated graphic here, but essentially, I think I can use this. Yes, I tried to highlight in the boxes what's most important. And so an individual is exposed to a certain allergen for a given concentration and for an amount of time. If they are sort of vulnerable, they will eventually, the antigen-presenting cells will pick up this allergen and then bring it into their system by the antigen-presenting cells. And this essentially, after quite a complex process, leads to, from these naive T cells, they get differentiated into Th2 cell phenotypes. And so that is what's most important, is that this Th2 differentiation of the cells is what is a hallmark feature of allergic diseases. And in this case, what happens is that the beta cells, because of all this, ends up producing antigen-specific IgE. So when this happens, an individual is said to be sensitized, and when they're exposed to allergens such as cats or house dust mites in the future, then they'll continue on to develop these sort of nasal symptoms. So stepping away from the biology, which wasn't really my focus, I looked a lot at the epidemiology of the disease. And so allergic rhinitis is a prevalent disease. Globally, about 8.5% of children aged 6 to 7 years old and 14.6% of teenagers have current symptoms. And this was established by the ISAC study that I'll be talking a little bit about later, but essentially they have data across the world on these sorts of prevalences. So we know that there is quite a prevalent disease and that the level of prevalence varies across the world, so it's not always the same. Interestingly, allergic rhinitis is also a dynamic disease. So here's the data on the first graph on the top, where I'm showing the prevalence of this outcome, or allergic rhinitis, over time. And this was done in the German Genie-Lisa birth cohorts that I work closely with. So we can see that as the kids get older, here I'm showing from 3 to 10, the prevalence of the condition also increases with age. There's also substantial comorbidity with other diseases. And this is a PDF or a graph that I've taken from a publication by the Swedish cohort that I work with. And you can see that the yellow bubble, which is rhinitis or allergic rhinitis, increases with time. And the overlap with eczema and asthma also differs depending on how old the child is. And so we know that it's prevalent and that there is substantial comorbidity with other conditions. And this sort of leads me to the main component of my thesis, which was looking at the risk factors for allergic rhinitis. So we know that it's prevalent, we know that it differs. So what is driving sort of the exacerbation, the prevalence of this condition? Well, there are risk factors which can be grouped as environmental risk factors that are present in one's environment. And the most important is certainly allergen exposure. So as I mentioned before, an individual needs to be exposed to the allergen in order to become sensitized if they are sort of to develop an allergic phenotype. And then subsequent exposure to this allergen will lead to the characteristic nasal symptoms. Which allergens are present in an environment differ depending on the climate as well as the vegetation present. So we know that climate has an influence on the allergens. We also sort of know with recent studies looking at changes in climate that climate may increase the length of the pollen season, which leads to longer duration of exposure, so more symptoms for a longer period of time. We also know from recent studies that the geographical distribution of allergens is changing based on climate change. There's certain studies which have looked at that idea. So we're getting new exposures or new allergens in areas that previously didn't have these types of allergens. So longer exposure, new exposures, and there's some evidence that shows that climate may actually change the pollen count, so we may be also seeing an increase in the severity of symptoms as well. In addition to climate, there's also been a lot of research looking at the effects of air pollution on allergens or allergic rhinitis. So air pollution may have direct effects on the individual, and this may be through interactions with the immune response, which may drive this Th2 dominated response, which leads to the allergic phenotype. Air pollutants may also lead to the generation of oxidative stress, which I'll talk about a little bit more in a future slide, and it also may increase airway inflammation. But air pollution may also not only affect the individual, but also the allergen itself, and this may sort of through indirect effects lead to change in health. And this may be by increasing the allergenicity or the morphology of the allergens so that we have different symptoms as a result of interactions between air pollutants and allergens. There's also a whole other side of the risk factors, which is the genetics. So we know because cases cluster within families, there's strong evidence that shows that an individual's genotype may affect the likelihood of them developing allergic rhinitis. And so far, there's been over 100 single nucleotide polymorphisms, or SNPs, which have been associated with this disease. And I try not to focus on genetics too much in this study, but essentially a SNP, or in this presentation, but essentially a SNP is just a change in the base pair. So we know that, if you remember way back in your undergrad, A binds with T and G binds with C. And so SNP can be a change in one of these base pairs, which can lead to downstream effects such as a new protein or differential binding for transcription sites. So essentially, a change in your base pair has been associated with allergic rhinitis. And one of the most recent GWASs, sort of with 6,000 cases, have identified 11 different genes which have been associated with this outcome. So we know that there's environmental risk factors, we know that there's genetic risk factors that we can identify, and if that wasn't complicated enough, we also know that there's gene-environment interactions. One of the most well-studied is the interaction between air pollution as well as some genetic phenotypes or some genetic polymorphisms, which may be mediated through the oxidative stress hypothesis that I mentioned earlier. And briefly, what this hypothesis states is that air pollution has been demonstrated to be able to generate oxidative stress in one's body. And so when you're exposed to air pollutants, your reactive oxidation species get produced. And usually, in an individual who's not susceptible, your body is able to curtail these using antioxidant defenses, and essentially nothing happens. But in individuals with possibly a diminished ability to metabolize these oxidative stress of a species may be at an increased risk of air pollution exposure. So in this genetic susceptible population may be more at risk of the adverse effects of air pollution leading to allergic rhinitis. And some of the potential genes that may be involved in this gene-environment interaction with air pollution have been identified, and some of sort of the key ones that we like to study are those involved in detoxification, such as GSTP1 or GSTM1, inflammation as a result of the Th2 phenotype, such as TNF, as well as the toll-like receptors which are involved in the immune response. So getting back to what I did during my PhD, essentially after months of thinking about it, I decided this was the aim of my PhD. It was to examine the influence of different environmental risk factors, focusing on traffic-related air pollution and climate, as well as genetic risk factors and any potential interactions between the two on the development of childhood allergic rhinitis. And as I said, I did this in 4 research objectives, which I will now briefly go through. So the first one focused really only on traffic-related air pollution. So I did a literature review, and we found that the experimental studies, so exposing people in sort of exposure chambers, like the work that Chris Carlston does at UGH, I believe, provide strong evidence of pollution-induced biological effects, especially for diesel exhaust particles. However, evidence from epidemiological studies, especially for non-asthma allergic phenotypes, was essentially deemed inadequate or insufficient to establish a causal association. So the tox studies are showing strong effects or consistent results, but the epidemiological studies are mostly inconsistent for non-asthma allergic phenotypes. So essentially in 2010, there was a call that more longitudinal studies are needed, and evidence from gene-environment interactions need to be considered. So this led me to my first objective then. I wanted to examine longitudinal associations between traffic-related air pollution and the development of asthma, allergic rhinitis, and aeroallergen sensitization in a birth cohort in Germany, and we had data available from birth to 10 years of age. And so here I'll just briefly go over the methods that I used. The study population that I had was the Genie Plus and Lisa Plus birth cohorts, which are 2 birth cohorts, but essentially they've been combined over time because they follow the same outcome and exposure assessment. And these cohorts are found in 3 areas in Germany. So one of the largest areas in the south, which contains the city of Munich, where Oktoberfest is, and that's also where I live. The one in the north near the Netherlands, it was called Wesel or Genie Lisa North, and then there's one in the eastern part of Germany in yellow. So we're able to look at sort of these associations in 3 different study areas. The outcomes that I considered was doctor-diagnosed asthma, allergic rhinitis, both of those separately, from 3 to 10 years, nose and eye symptoms at 3 time points, and we also had objective measures of sensitization available at 2 time points. The predictors that I looked at were predictors for traffic-related air pollution primarily, so we used NO2, PM2.5 mass, and PM2.5 absorbance, which were all indicators of traffic-related air pollution, and these were estimated at the individual level using land use regression model. So we're able, based on a model that we developed, to estimate the air pollutant levels at each child's home address. We also were able to assign ozone estimates, which more, based on the model we developed, is more representing air pollution in general and not necessarily from traffic-related sources. The analysis that I did, because we had data over time, was a longitudinal analysis using generalized estimating equations. And importantly, and this will be a theme that comes up over my thesis or over the next this presentation that I'm giving, is that I was interested in looking at not only the pooled associations when you pool it all together, but also looking at area-specific associations to look for consistency across trends. So I'll be building on this slide. First, just to give you an idea of how it's built, on your left is the total population. So this is the pooled data when I pull it all together. And the different colors represent the different outcomes that I looked at, with asthma in red, allergic rhinitis in purple, eyes and nose symptoms in blue, and sensitization in black. So the total population is on the left, and then I show the associations for the 3 different study areas. And you can see that these are the results for NO2. We also looked at the 3 other different pollutants. And I realized that this is a lot of dots on a graph, so I'll try and attract your attention to some of the most important results. For example, in Genie Lisa South, which is in the southern part where it includes the city of Munich, we found that certain air pollutants appeared to be actually negatively associated with outcomes. So taken at face value, this seems to indicate that air pollution may actually be good for you if you live in Munich. However, we also looked at different areas, so in the north as well as in the east of Germany, and different pollutants, and we found that other outcomes, or these same outcomes in the different areas, seem to be positively associated with air pollutants. So what we were hoping to do is get a consistent picture across the study areas, across the pollutants, and across the outcomes. And we don't see that here in any of the analyses that we did. And essentially, the conclusions that we came to for this analysis was that there are no consistent associations with traffic-related air pollution and the outcomes that we considered that seem to replicate in the different study areas. And this is important because we're using the same standardized outcome and exposure assessment. So the idea is that there isn't any consistency, which sort of reflects the literature in the past, that although you can find one association here and one association there, you can't find it consistently across the study areas. And in fact, that was our most important conclusions, was that heterogeneous results were observed across the different study areas. And I won't go into all of the sensitivity analyses that we conducted, but you can imagine that we tried pretty hard to get rid of these protective effects in the south, and these results were quite consistent no matter what we did, the movers, or if we established or assessed the air pollutants at the different ages, so birth, 6, and 10 years. So these were actually quite robust. So we focused on this. We see heterogeneous results across the 3 study areas. We essentially needed more data to see if this was also applicable in different areas. And what about considering gene-environment interactions? Which led me to my second objective. So in this study, we managed to look at sort of similar associations, so between traffic-related air pollution and allergic rhinitis air allergen sensitization, but this time we were able to recruit data from 6 birth cohorts. So the 2 from the past and then 4 additional birth cohorts, which I'll introduce. And we also had available data on 10 SNPs, so 10 base pair changes, in genes involved in detoxification, inflammation, as well as 2 of the toll-like receptors. And these SNPs were selected based on first data availability within the cohort, but also because of the literature. So there have been interactions, especially with asthma, associated with these different SNPs. So this is the TAG study population, which is essentially a collaboration that was led by Mike Brower and Chris Carlston from this school, where we combined data from 6 different birth cohorts, 2 of which are from Canada. You may recognize CAPS in the second row and SAGE at the bottom, which are Canadian-based. And then we had a birth cohort from Sweden, Bamsa, the same 2 German cohorts, so Jeannie and Lisa, and as well as a Dutch cohort. And this is a lot of information, so essentially what you should take away from this is that they were all population-based to start with, except for CAPS, which was a high-risk cohort. So the children were selected based on a positive history, a family history. The recruitment was all essentially in the mid to late 1990s, and the total combined population was just over 15,000 children. This is all of the children that had either 1 outcome or 1 air pollutant. So when I show sort of specific combinations focusing on only air pollutants or only certain SNPs, we essentially had analyses ranging from between 4 to 8 to 9,000 children. So still the largest gene-environment interaction study for allergic rhinitis to date. And another important consideration is that the allergic rhinitis definition, although quite consistent, did differ across the cohorts. For the first one, it was based on a symptoms or diagnosis of allergic rhinitis. For 4 of them, it was on a diagnosis, or a doctor diagnosis of allergic rhinitis or hay fever, depending, and these are from parental completed questionnaires, all questionnaire-derived data. And the Dutch cohort was only based on symptoms, and so there was a little bit of heterogeneity in how allergic rhinitis was defined. So back to the methods. So the study population are these 6 birth cohorts. The outcomes that we considered, we only looked at 1 time point because it was the easiest to collect data from all the birth cohort, and they did not all have sort of longitudinal data available. So we had allergic rhinitis at either 7 or 8 years, as well as air allergen sensitization at 7 to 8 years, depending on the cohort. The predictors that we used were the same as in the last one, so land use regression-derived estimates. This time only to the birth address were available for all of the different cohorts. We also looked again at these 10 SNPs involved in genes hypothesized to potentially play a role in the gene-environment interactions. The statistical analysis that we did was a cross-sectional analysis, again focusing on the pooled as well as, importantly, the cohort-specific associations. So this is just to give you an idea of the type of data that we were dealing with. So now I'm showing the NO2 distribution, so the values of NO2 levels at the home address at birth across the different areas, and the different colors essentially mean the countries. So Netherlands is in purple, for example. And what's interesting about this is not only did we increase the number of children that we had in TAG, so looking at all of the cohorts, but we also increased the exposure range that we were able to look at the different associations across. So we sort of gained statistical power in a variety of ways by combining these data. So I'll only show select results. So this is only focusing on the air pollutants. So just forget genetics. So I'm only showing associations with the air pollutants and related to in association with allergic rhinitis. And what we can see from NO2 is that, consistent with what we had found before, we essentially see absolutely no association between NO2 at birth and allergic rhinitis. And this is more or less, if you look across the different cohorts, what we would expect if we had looked at any one of these cohorts individually. However, when we looked at other pollutants, such as PM2.5, we did find a statistically significant increase, this time in the right direction, which was also nice to see. But we found that it was entirely dependent on certain cohorts. So in this case, that association is entirely driven by the Piyama cohort. And we were really interested in looking across cohorts. It was important to keep these sort of outlier cohorts in mind. And we don't really understand why it may be elevated in only certain areas. It might be due to the different types of air pollution sources there or the population, but we don't have data to be able to explore that. So it's more or less speculation as to why that association is only visible in the Netherlands. And these, when we looked at gene and air pollutant interactions, when we considered the genetics and sort of stratified by the different SNPs that were available, we found that we did not reveal a genetically vulnerable subgroup for any of the air pollutants. And when we tested specifically for gene-environment interactions in the cohorts, in the models, they were all non-significant. So the null results that we were seeing did not seem to depend on the genotypes that we considered. Now, if we forget air pollutants, we were also interested to see if there might be any main genetic effects. So are the children at risk based on genotype, regardless of their level of air pollutant exposure? And what we found from the 10 SNPs that we considered, there were 2, one of which was in the TNF gene related to inflammation, and one of which was in the TLR4 gene, were significantly associated with allergic rhinitis. And these results were robust based on several sensitivity analyses. So for example, in the last one, if we excluded SAGE, the sort of outlier there, the estimate, if not remained statistically significant, it was quite consistent. Sometimes the bands got a little wider because we lost some kids, but the results were quite consistent for these 2 SNPs. And so to summarize for objective 2, consistent with what we had found before, we don't find any consistent evidence for air pollution effects. And this is now including 2 cohorts in Canada and 4 in Europe. So that's quite a lot of children. We're not finding anything. And these null findings are not, they do not seem to change based on the genotype. So we're not able to identify any gene-environment interactions. However, we were able to identify 2 SNPs, the TNF and TLR4, which may be risk factors for allergic rhinitis at the school age. And what was interesting is that these genes are also associated with asthma. And so we started to think a little bit more about what about the genetics behind what's going on. So we know that there's a well-established comorbidity between allergic rhinitis and asthma, but we don't know if this is because one causes the other, so allergic rhinitis leads to asthma or vice versa, or because maybe they have common environmental and genetic risk factors. So I'd already shown that we did have common genetic risk factors. And so we sort of focused on what's the biggest risk factor we can find for asthma, and this is the 17q21 locus. And so we don't necessarily need to understand what that means, but it's essentially an area on the chromosome 17, which has been quite robustly associated with asthma. It was the first, it was found in the first GWAS for asthma, and it's been shown repeatedly that this is a strong risk factor for specifically childhood asthma. And when we looked at whether this may also be associated with allergic rhinitis, we found that a few studies had looked at it beforehand when they were smaller and the smaller GWASs, but essentially there had been conflicting literature, and the last 2 studies were showing an association. So we said, okay, we have this data. Why don't we make this objective 3 to examine whether 7 SNPs located at this asthma risk locus are also associated with our cohort or with allergic rhinitis in our cohorts. And we're also interested to see if any possible association may be modified in asthmatics, because we know that this risk locus is also associated with asthma, and that there's an association between asthma and allergic rhinitis. The study population that we looked at were the same 6 birth cohorts in TAG, so we'd already established this sort of international collaboration where they had genetic and environment data, so we took advantage of that. And you see that the sample size here is a little bit smaller, and this was because of the available genetic data. And so the SNPs for this area were based on imputed genome-wide data, which was available for a much smaller subset of the cohorts. The outcomes that we looked at was allergic rhinitis as defined previously, as well as concomitant aeroallergen sensitization. We really wanted to get at the cases that were sensitized right now, or that had a positive test for sensitization and allergic rhinitis, so that we had a very specific phenotype to look at for the genetic risk factors. The predictors that we looked at were 7 SNPs in linkage disequilibrium that were at this particular locus. And what this essentially means is that these SNPs tend to travel together when the chromosomes undergo recombination. So it's not that we're looking at 7 independent SNPs that sort of travel as they will, but rather that they are an LD, and so they tend to move together. And so the associations that I'll be showing are essentially like 7 similar hits. And the statistical analysis that we looked at was a longitudinal analysis using generalized estimating equations, and again focusing on pooled and cohort-specific associations. So I'll be building upon this graph, so it's important that we sort of understand the structure. So we have here is the 7 SNPs along the bottom, which are in LD, so they are correlated. And the letter is just the effect allele that I tested. I could have tested the other one, and then all of the SNPs would essentially be flipped. So instead of being protective for the first one, it would be detrimental. And the reason that I present in this way with some SNPs being actually protective is in order to maintain this sort of haplotype pattern. So this is how these SNPs tend to be inherited together. What I'm showing here now is the associations with just allergic rhinitis, as what defined as having allergic rhinitis and being sensitized. And what we find is that 6 of the SNPs that we looked at are indeed associated with allergic rhinitis. When we looked at these associations with asthma, we found very similar associations, which was a relief because this area, this genetic variability, has been frequently associated with asthma. So we would have expected to see these significant associations. So that was nice. What we hadn't been looked at in this context is whether, when you modeled allergic rhinitis and asthma, you may also get the hits. And here we see that the effect estimates, although the confidence intervals do overlap, tend to be a little bit greater, a little bit further away from the null. So this may be indicative that genetic variability at this locus may be associated with the development of multiple clinical phenotypes or a more severe type of disease. But what about when we take away asthma? And so these are children that only have allergic rhinitis, but that weren't diagnosed with asthma at that same time point. And we were interested to see that at least a few of the SNPs, I think 5 of the 7, were either borderline or statistically significant with genetic variability at this region. However, these last dots, these purple dots, don't address whether the child may have had asthma earlier on. These are just children with allergic rhinitis who don't have asthma right now at this same time point, but they may have had asthma when they were younger. So we did a stratified analysis where we looked at allergic rhinitis associations with the genetic variability, but now stratified by whether or not these children had early life asthma. And we can see that the black dots, so the children who had asthma as a child, there is an association between allergic rhinitis and the different SNPs. But when you take asthma out of the equation, which is the red dots, so these are associations between genetic variability and allergic rhinitis, but amongst children who've never had asthma, you can see there's essentially no association, which sort of indicates that asthma has to be in some way or another playing a role in this association. So to summarize, genetic variability at this region was associated with allergic rhinitis, and the association seemed to be most pronounced among those who were also diagnosed with asthma. The stratified analyses that we conduct suggest that the association between allergic rhinitis and this genetic region is likely mediated or supported through an asthma-dependent mechanism. Okay, so I've talked a lot about air pollution, I've talked a lot about genetics, and I've talked a lot about birth cohorts. So now we can switch gears to climate, which I think is actually maybe one of my favorite parts of my PhD. And so we know that changes in climate can alter allergens. It's well documented so that the climatic conditions affect the vegetation in an area. And we know that allergens are the primary risk factors for allergic rhinitis. However, studies which have looked at sort of the broader picture, looking at changes in climate, which likely lead to changes in allergens and then to symptom prevalence, are essentially conflicting and few. There have actually, and this may be because the influence of climate on the allergens is likely regional dependent. So you can imagine that an area which has a lot of changes in climate will likely lead to different vegetation, whereas an area which has only small changes of climate will have a similar vegetation in that area. And so there was only 2 studies which had looked at multi-country data, and this might give us sort of a more complete picture of the relationship between climate changes and how this may affect allergic rhinitis prevalence. And so we wanted to look at these multi-country associations, but we asked, can we do this on a global scale? And so my last hypothesis, or my last objective, was to assess associations between two types of rhinitis, so intermittent persistent rhinitis, with climatic factors and vegetation in a global context. And here's where I introduce the international study of asthma and allergies in childhood. And so I think anyone who works in allergies is quite familiar with this study, but it's essentially the largest research collaboration in the world, and they've gone out and done questionnaires on 13 to 14-year-olds and 6 to 7-year-olds to assess the prevalences of asthma, allergic rhinitis, as well as eczema. And these questionnaires are quite standardized, and they've been translated into a variety of different countries, languages for the different countries. And the data that I was using was from phase 3. And this involved data from 222 centers in 94 countries for their older age group. And to give you an idea, I believe this was just over 200,000 children. So this is quite a large sample size. But we do group them into centers. So I'm not looking at an individual, but rather if you take Vancouver, and as an example, I'm looking at what is the percentage of children with rhinitis symptoms in Vancouver. And we define two outcomes. So intermittent rhinitis, which is when a child reported rhinitis at least 1 month during the year, but never for 2 consecutive months. Or persistent rhinitis, which was reports for at least 2 consecutive months. And then we calculated the prevalence of these children in a given center. Yeah. And this is just a map to show you of the level of the data that we had. So here I'm showing the prevalence of intermittent rhinitis for the 13 to 14-year-olds, so data on 222 centers. And you can see that we had good representation from Europe, as well as South America and some of the few islands in the oceans. And what's also important to take from this is that there is, first of all, good geographical distribution across the world, and also that there seems to be differences in prevalences, which may be due to changes in environmental factors such as climate. So the predictors that we looked at were 3 climatic factors, so temperature, precipitation, and vapor pressure, as well as we attempted to look at changes in vegetation. And this is using this increasingly popular NDVI index, the Normalized Difference Vegetation Index, which essentially we derive from satellite data, and it's an index that runs from 0 to 1, and the greater the number, the closer you are to 1, the greener your environment. And we use data across years to look at the monthly mean, minimum, maximum, the range, as well as the standard deviation of monthly measurements for this given area. The statistical analysis that we did was an ecological analysis because I was using center as the unit of analysis and not individual. And we ran a linear regression mixed model assessing for center and country level effects. And so I'm going to try and explain this with a few examples. So this is the associations between country. So now I'm comparing sort of Canada to Germany to China, and those sort of between country associations when they're related to temperature, precipitation, vapor pressure, and vegetation, which is indicated by the different colors. So if we just focus on the first 5 dots, which are red, focusing on temperature, we have significant positive associations between the mean and the minimum monthly temperature. So for taking the first dot, if you have a mean, if your country has a mean warm temperature, then you have an increased risk of the country level prevalence of this disease. If you look, for example, at the max and the min, so if you have a large range, such as in Canada, where we get really, really hot and really, really cold, then you have a lower change in your prevalence of intermittent symptoms. So what's important to take from this is not necessarily that one goes up and one goes down, but that climatic effects, temperature, precipitation, vapor pressure especially, seem to be associated with the country level prevalence of an intermittent rhinitis. When we replicated this looking at persistent symptoms, we found no associations with the climatic factors, and none of the 2 conditions that we considered were associated with vegetation. So these are the between country associations, comparing countries. For a few of the countries that are involved in ISAC, we're also able to look at within country associations. So these are the countries that participated greater than 1 center. So you can imagine before I was comparing Canada to Germany to the Netherlands, and now I'm looking at within Canada, so Vancouver, Montreal, Toronto. So these are the within country associations, and we can find that for intermittent symptoms, the first graph, Temperature and vapor pressure seem to be associated, but there wasn't quite as many as when we were looking between countries. For persistent symptoms, where we found no associations in the first analysis, we do find that at least temperature and vapor pressure may be associated with persistent symptoms when we look at within the country, so comparing actual centers within a country. So I'm sure I lost a few people, but essentially to summarize, there is substantial variation in intermittent and persistent rhinitis prevalence. So that was already sort of a step forward in this analysis because they had never established these two outcomes in ISAC. So hopefully they'll be able to use these and continue looking forward with the different associations that they consider. And we did find several between and within country spatial associations with the climatic factors that we considered. Unfortunately, we weren't able to identify any associations with vegetation using the NDVI index that we considered, and I'll talk a little bit more about that in the limitations section, which I am now at. So there were several strengths and several sort of limitations in the project that I did. Focusing on the birth cohort data. So this is objectives 1 to 3 when I looked at traffic genetics using the birth cohorts. First of all, it was a huge strength that all of these were indeed birth cohorts, where there was prospective, repeated outcome, and covariate data collection, which does reduce the level of recall bias and other biases that may be introduced. The air pollution assessment was at an individual level, so we were able to estimate the air pollutant levels right at their home address, but this is restricted to the home address. So we have no information on the air pollution levels at home, during commuting, as well as indoor environments. The selection of the genetic risk factors that I looked at, we followed a hypothesis testing approach. So we looked in the literature, which ones we would expect or which ones have been identified, but it's certainly far from being comprehensive. So there's definitely additional genetic risk factors that need to be considered, as well as gene-environment interactions, and going even a bit further than just looking at SNPs, but also looking at epigenetics as well as gene-gene interactions. So there's much more here to be done. An important strength of the data, of the analyses that I did do, was that we pooled several sources of data, not only from Canada, but also internationally. So this allowed us to increase our power, and we were able to look for consistency across associations. So you can imagine that if we had looked only in the Netherlands in that first project, or in certain areas within Germany, we would have found statistical associations. But when we have data from different areas, we're really able to look consistently across to see if we can find a trend that seems to be true in more than just one area. But of course, these cohorts are not identical, and so it was important to consider heterogeneity in terms of not only the outcome assessment, but also the different types of pollutants that may be present. Then focusing on ISAC data, which is sort of this last global study that we did, which is quite different. First of all, an important strength is that we were able to establish 2 new rhinitis outcome definitions. And the reason that I didn't invent these definitions, they're being newly introduced to replace seasonal and yearly allergens, which you can imagine are quite difficult to establish on a global basis. So seasonality in Canada is quite different from seasonality in Brazil. And so using these new outcome definitions, they're based on the duration of symptoms, which is applicable worldwide and actually better reflects what an individual is actually experiencing. Because of the structure of the data, we are able to examine both center and country level associations, which to some extent appear to possibly have different effects. And we were able to replicate the associations in the younger age group. So everything that I showed was only based on the 13 to 14-year-olds, but we also had these data available for a smaller subset, I believe it was 100 and something centers in the younger age group, and all but one of the significant associations were in the same direction. So they weren't all statistically significant, but we did have fewer centers, both in terms of the number and the range of exposures, but the direction of the effects seemed to be going in the same way. An important strength is that it was worldwide coverage, so we were able to make this map, which I think is always fun, but it is cross-sectional, so we're not able to look at temporal associations, or the temporality of these associations, and it is an ecological study design. So we're not able to confirm that the associations that we see at the group level also hold down to the individual level. And the environmental assessment for this study was, I think, pretty good for the climatic variables. So when we assign a climate, or the level of climate, to a certain center, if it's hot in Vancouver, it's generally hot everywhere in Vancouver in that area. So the effect of the climate variable is quite stable over a certain distance, where the NDVI measures an idea of vegetation. And it is possible that vegetation may vary more considerably over smaller distances. So if it's quite green in Stanley Park, it doesn't mean that it's really quite green a little bit further down. And the NDVI index also only gives us an idea of how green it is, but it doesn't allow us to look at what type of plants they are and what type of allergens they may be releasing. So after all that, I'd like everyone to recall the aim, which was to look at environmental risk factors, focusing on traffic-related air pollution and climate, as well as genetic risk factors and potential interactions on the development of childhood allergic rhinitis. So after lots of thought and all the analyses that I conducted, I was essentially able to come up with 3 statements about the type of research that I had done. First of all, traffic-related air pollution was not consistently associated with allergic rhinitis. And this was true looking specifically within Germany at 3 study areas where we had the ability to do a lot of sensitivity analysis, and then again looking at data from 6 birth cohorts from within Canada and Europe. And these null associations were also not modified by any of the gene-environment interactions that we tested. However, we were able to identify several genetic risk factors that were associated with allergic rhinitis. So these were the TLR4, the TNF gene, as well as this asthma risk locus. And all of these genes are also associated with asthma, which adds evidence to the idea that there may be common genetic risk factors between allergic rhinitis and asthma. And finally, future changes in climate may alter allergic rhinitis prevalences. And this is based on the last study that we did. We say may, and with caution, as it's only the 2nd global study to do this. The first was based on phase 1 in ISAC. So these types of analyses need to be replicated both in terms of smaller studies as well as looking globally to see what any effect of future climate change may have on allergic disease prevalences. So I'd like to finish by acknowledging my committee, Mike and Chris, who were my co-supervisors from SPPH, as well as Joachim Heinrich, who is essentially my boss in Germany, that I work with in Munich, and Andrew Sanford, who is also on my committee. I'd also like to specifically thank all the TAG and ISAC collaborators for giving me access to their data, as well as their time and all of their friendly suggestions. And I'd also like to thank specifically St. George's in the UK, Karolinska in Sweden, and Helmholtz in Germany, which all welcomed me into their teams at some point in time during my PhD to conduct this research for a variety of a few days, a few weeks, and now I've been in Munich for a few years. That's all. Thank you.
Online, okay. Fantastic. That was great to hear about that research. Okay, opening up for questions from the floor.
Thanks, Elaine. Very interesting. Just curious, did you look at international data on childhood allergic rhinitis and asthma in consistently high pollution countries like China, Pakistan, India? And also, I was curious if you could pull up that global map where there were various extents of morbidity. Yeah. So just looking at the Southeast Asia portion, not a lot of red dots or high prevalence of rhinitis. Just wondering if you had looked at any international data to compare.
In relation to air pollution?
TRAP and
Yep.
allergic rhinitis.
So I specifically did not look at this in relation to air pollution, but Ross Anderson, who is actually one of the co-authors on this study, specifically looked at satellite-derived air pollution levels. So they use satellites to derive an air pollution estimate to the to the center. Did look at this in terms of rhinitis, asthma, and another outcome, and then specifically also at severe asthma. And he found either null or protective associations in the case of severe asthma. So again, not saying that air pollution is good for you, but it's hard to control for the level of residual confounding in these analyses. And also, one of the reasons we looked at climate is that, when it's hot in Vancouver, it's hot in Vancouver. But the air pollution estimates, you have a lot of within city variability that may actually be more important in terms of exposures than between city variability. So they have looked at it. I didn't do this as part of my PhD.
Thank you for the talk. I was just wondering if you could talk a little bit about the application of this work, the implications of it, and also the next steps. So what do you see this work leading to? Are there any changes that you see happening as a result of it? I know that you didn't necessarily find what you were hoping to find or look for, but or maybe what the next steps are with the work.
Sure, that's a great question. It's a defense question, actually. In terms of the air pollution, what we did, it was somewhat of a disappointment. It would have been nice to see strong effects and then gene-environment interactions as well. That would have been perfect. We do see that for asthma. So a side project that was done, led by Elaine McIntyre, who was the postdoc on this, did consider asthma, and they did identify gene-environment interactions. And it sort of seems to be consistent in the literature that the associations with asthma are much stronger than they are for other phenotypes, such as allergic rhinitis. In terms of the future work of the genetic risk factors, there are quite a few studies which are looking a lot on the 17q21 locus to try and identify what it is this locus is doing. And so at this point, for allergic rhinitis, it's quite novel, but for asthma, it's not. And so I think the future work, not work that I will be doing, but will be to sort of identify what these SNPs are doing or what this locus is actually doing, so that we know, for example, for the TNF, that the SNP that I showed changes the promoter binding site, which then results in a decrease in the inflammatory protein, or an increase. And so there is some work, but it's also sort of extending it past just looking at one SNP, but maybe looking at a combination of SNPs, which was sort of a first step gene environment approach. In terms of the climate data, I think we need a few more years to allow the climate to change, so that we can look, what would be really interesting would be to look at temporal changes. So now we have this sort of baseline analysis, and when climate does change in a while, we'll be able to see if there's any changes that also follow it, rather than just looking cross-sectionally, but also temporally.
Hi, thank you. I know most of your work concentrated on data from Western and North American countries. Do you think that that data is of, especially the environmental air sampling quality, is of good quality? And how would you compare it to the data coming out of developing countries, China, India, places like that?
So the reasons that we picked these cohorts to be part of TAG, these 6, was based on the availability of individual level data, as well as the genetics. So we would have loved, I would have loved to have sort of a cohort in China or something like that, but they didn't have the requirements that were needed to participate. The air pollution estimates that we did have for the home address are actually quite good. These are quite established models, and they've been validated with personal or like actually monitoring. So like the level may not be exact, but the spatial distribution, which is what you're really looking at, is comparing spatial associations, is quite good. And this has been extended here in the air pollution work as part of the ESCAPE project, which was sort of this Europe-wide project which developed similar models in 37 countries there. So I'm pretty confident that the air pollution that we had was pretty good for what we were looking for. Your question relating to the developing countries was the level. I mean, the levels are clearly much higher there. And so I think it would be really interesting to look there, as well as considering the indoor air pollutants, I think are quite an important factor there that need to be considered in any association with outdoor.
Questions? Last chance? Nothing online? Well, with that, I would like to thank you very much for a fabulous seminar, and all best wishes for your future work.
Thank you.